Welcome to the breakout session 5. Out of respect for our presenter, please silence your cell phones. If you must take a call, please step out of the room. If you want to receive CCA credits for the session, you must sign the sheet on the table in the upper lobby. Please hold all the questions until the end of the session. We will end on time, so if you would please clear the room at the end of the session. If you have questions for the speaker, please go to the cherry room following the session. To the pressing worldwide challenges of restoring topsoil, soil ecologists, Dr. Christine Jones offer an accessible, inspiring perspective. Over several decades, Christine has worked with innovative farmers and ranchers implementing regenerative land management practices that enhance biodiversity, nutrient cycling, carbon sequestration, productivity, water quality, and community and catchment health. Following a highly respected career in the public sector research and development, Christine founded Amazing Carbon and organized a series of managing the carbon cycle forums to promote the benefit of soil carbon. In recent years, she has gained an international recognition as a speaker presenting at workshops, field days, seminars, and conferences throughout Australia, New Zealand, South Africa, Western Europe, Central America, USA, and Canada on carbon and nitrogen dynamics in agricultural systems. The titles of Christine's presentation is Soil Community Tipping Points, Enhancing Nutrition Yield and Resilience Through Quorum Sensing. Please welcome Christine.
Yeah, so what I want to talk to you about today is probably something that's going to be a little bit new for some of you, and we're looking at what is it that's creating some, what is it behind some of the absolutely extraordinary changes we're seeing, where people reach a certain level of diversity in their cover crops or in their forage crops, and we're seeing some really amazing changes in their soils, in their animals, and I want to just show you some of those changes and then talk about what might possibly be behind that. Now, last year there was a scientific report was published, which was a census of life on Earth measured in gigatons. And this is just a little bit of background information for you to set the scene for what it is that I want to talk about. And they found that there was 550 gigatons of carbon-based life forms on Earth. And when they broke that down, 450 gigatons of that 550 was plants, which is not surprising. Many areas of the world that you go to, not so much here in Kansas, but when you look around, you see an awful lot of plants. So the most biomass, the most living biomass on Earth is actually in the form of plants. But what I was interested in is, well, what makes up the other 100 gigatons? You know, where do we sit, where do our plants sit, and our soil biology sit in the 100 gigatons? And what we found is that little things that we can't see, the protists, the archaea, the fungi, the bacteria, they actually comprise 93% of that remaining 100 gigatons. So we've got our plants, and then we've got all these little things that we can't see are actually making up the majority of biomass on Earth. If we look at this diagrammatically, in that diagram there you can see you've got the protists on the top, the orange bit, then the archaea, the purple bit, then fungi, and then right at the bottom there, bacteria, 70 gigatons. We can't see them, but they're a very potent force on this planet, in our soils, in our plants, in our animals, and now we're finding in the human gut. Very, very important. There's far more of them than we ever realised. And things that we can see, like insects, mollusks, things like snails, or in the ocean you'd have like all your shellfish, and then other things in the ocean like the fish. I mean, think how much of the land, of the area of this planet is taken up by oceans, and all the life in the sea, and all the things that we can see above ground, your livestock, the birds, everything. That makes up 7% of that 100 gigatons. So that's not even 7% of the total, that's 7% of that 100 gigatons. And humans, well, we're quite insignificant in the overall scheme of things, apart from the fact that we've made a huge mess of this planet. We only constitute 0.01% of the biomass of life on Earth. So we've pretty much punched above our weight when it comes to the changes that we've made. And if we go back to this original graph that I showed you, humans wouldn't even feature on there because we weigh, collectively, all of us, weigh 0.06 gigatons. So we don't even register on that graph. There's more protists than there are humans. So the point of showing you that and the biomass of microbial life, of microscopic life on this planet, is that we are embedded in this microbial world. And for those of you who listened to David and Anne's introductory speech yesterday, and Anne was talking about, the microbes in our gut, a human gut microbiome, and how that all works and how important that is for our health. We also have a microbial world embedded within us. So do our animals and so do our plants. And what I'm going to be talking about today is how our genes, the genes in our bodies, in our animals' bodies and in plants' bodies, can actually be activated. They can either be switched on or switched off by those microbes, and how they do that, because this is very important for us to know in terms of animal production and plant production, is what is actually going on. And what we're realising is that we used to think, like we might think of ourselves as a discrete unit, or an animal or a plant as a discrete unit. Actually, we're composed of interdependent communities of organisms. All of us are. We're not only dependent on each other, but we're dependent on everything that lives in us and on us and around us, and so are our animals and plants. Because there isn't any such thing as an independent organism on this planet. Every species needs other species in order to survive. If we were to eliminate everything else that lived on this planet, which we seem to be doing a pretty good job of attempting, we would not survive. And if we eliminated everything that lived in us, we would not survive. And the same goes for our plants. So if we look at human health, we're now realising, I don't know how many of you heard David Johnson's talk, or what Anne was talking about the other day, that many of our current health issues are actually linked to a failure to support a diversity of microbes in the human gut. And that's the important point there, is the diversity of them. And the American Gut Project, which is a citizen science project in which they collected 11,000 fecal samples from people all across the United States. People just sent those in the mail, and which was interesting. I presumably they all got there intact. And they found, and as well as sending a sample, you also had to complete a questionnaire on what you ate during that week and how your health was. There's a whole lot of questions about do you have any autoimmune disorders or other health issues, and what were you eating. And what they found was that people who ate at least 30 different kinds of plant foods in a week, it didn't matter whether you ate meat or not, you could eat lots of meat provided you had lots of plants as well. They were far more healthier, and they had more, because they had more diverse gut microbiomes. People who ate 10 or less different kinds of plant foods in a week had all kinds of health issues. And if you look at the standard American diet, the SAD diet, you find that there's 5 major food groups that people consume in this country, and you probably had most of them for lunch. It's pretty sad when you see the low diversity of food that's offered in this country and the low diversity of food that people consume. And they probably eat the same 5 things every day, 7 days of the week. So you're not getting enough diversity in your diet to actually support a gut microbiome that's going to make you healthy or protect you from autoimmune diseases like cancer, diabetes, Alzheimer's, rheumatoid arthritis, Crohn's disease. I mean, the list goes on and on and on. Psoriasis, all those kinds of things. They're becoming more and more common in the population. So if we look at animal health, Fred Provenza has done quite a lot of work in this, and he says that livestock that consume diets rich in plant secondary compounds, this increases again the microbial diversity in their gut, gives them the ability to digest a wide variety of feeds, and therefore improves their feed conversion efficiency, which is very important if you're making money out of growing livestock, and definitely improves their immune function. So we know this in humans, we know this in animals. Does this same diversity principle actually apply to soils? If we have a more diverse microbiome in the soil, is it going to lead to more productivity on the farm? And what we've discovered through research is that the susceptibility to pests and diseases, low nutrient status in plants, and poor plant productivity are definitely linked to low diversity in the soil microbiome. So it doesn't matter whether we're talking about animals, plants or people, if it's a simplified system, if we actually reduce the diversity of life in that system, it is going to be a degraded system. There is no way it can function at its optimum capacity if it's simplified. We want to we don't want to have all these complicating factors. We keep reducing things, and then it doesn't work and we wonder why. Most agricultural landscapes have become so simplified that we're not even aware of how incredibly productive a more diverse system could be. And if you think about the American prairies, which was mostly the land that was originally used for growing crops, there would have been 500 to 700 plant species in a relatively small area on those prairies. About 40% grasses and 60% forbs. There are still some ranches around that have over 700 different ground cover plants in a relatively small area here in the United States. So we're assuming that that's what the number would have been. It could have even been higher. Now you take that number of plants all growing together and think about the diversity of life that would have been in the soil under those plants, and then you go to, okay, I'm just going to grow corn, or I'm just going to grow wheat, I'm just going to grow soybeans, and think just how simplified that landscape is. So how can that soil microbiome function? And we know that a simplified microbiome is dysfunctional. It doesn't matter what you pour on there. We can try and put on all the fertilisers, all the NPK, whatever that you like, or put a bit biostimulant on, but it's still not diversified. You're not going to get anything like what you could get if you had a much wider range of microbes in that soil. We know that a diverse system is more productive, so the original American prairies produced far more than we can possibly produce with a monoculture on that same area of land. That's always been put down to what they call niche complementarity. In other words, if you look at the architecture of the roots on those plants, some have deep tap roots, some have shallower but much more fibrous roots, and that they're all occupying different parts of the soil. And it was thought, well, if you just have one kind of plant and one kind of root system, then they're competing to some extent, whereas these are more complementary. And that was the one reason that was given for a community of different kinds of plants to be able to be more productive. We now know, probably 10, 20 years ago, we realised that these plants are all connected underground with mycorrhizal hyphae so that they form common mycorrhizal networks. We know they can exchange nutrients, water, carbon, energy flows, and they can keep each other alive. So that we knew that microbes were involved to some extent in keeping that plant community more productive. Now that we've discovered even more about how these microbes work, we realise there's a lot more to it than that. But if we just look under a plant community like this, even just at the fungi that under there, for example, these are fungal spores from a diverse plant community. You can see there's lots of different kinds of fungi there. And because we never see these with the naked eye, we're not realising just how extraordinary this underground world really is. So my question about the soil microbiome is, does it respond differently once a certain threshold of plant diversity has been achieved? And my reason for asking that question is because I've seen some, what I believe to be tipping points in terms of the whole system changes completely once we get over a certain number of plants. And the first evidence that we saw of this was in some trials that were conducted by the Burleigh County Soil Conservation District. Most of you would have listened to Jay speaking here in this room a little earlier today, and he was one of the people that was involved in these crop demonstration trials. They had been here at No-Till on the Plains and heard Adamir Caligari say, oh, you can have all the cover crops you like, but if they're not diverse, you're not going to get very far. You really have to have diversity in that mix. So they went back to North Dakota and decided to run a trial where they had some monocultures. So they planted individual plants that had been used as cover crops, oilseed radish, purple top turnip, soybeans, red clover, ryegrass, etc., in 1 acre plots. So they had 8 different species that were planted individually, and then they had some multi-species combinations as well. And what happened in that year was it was an incredibly dry year. They got 1 inch of rain between the seeding in late May and the harvest in late July. And so Jay was thinking, well, this has just failed. Nothing is really growing. All of their monocultures look like this. So this is the oilseed radish. This is at the end of July. And everything else, the ryegrass, the soybeans, the cowpeas, everything that was grown on its own looked like that. There was virtually nothing to harvest. But where they had put a multi-species mix in, so where they had cowpeas, soybeans, radish, millet, sunflower all growing in together on the 31st of July, the same day, this is what the multi-species crop looked like. So how can it be so green and so productive with all those different plants growing together? So this is a research trial. It's not anecdotal. They've got all the data. These crops were harvested. They've got the dry matter information and protein contents and energy contents, and everything's all been done on these plants. You know, how can we go from monocultures looking like that, which is fair enough on an inch of rain, and then a polyculture looking like that? But the 3-way mix, the 4-way mix, the 5-way mix didn't do this. They all failed. We had to get up to this, having a certain number of plants in a mix for it. Suddenly it's a tipping point and everything changes. And that's what's interesting me. What are these tipping points and how do we find them? The Chinook Applied Research Association in Alberta, in Canada. I was there in July 2015. Again, a very, very dry year. They had been convinced to try a cocktail crop because Gabe Brown had been up there speaking to them about cocktail crops. And so they had a field where they'd just planted triticale, and it was obviously not going to go through to yield. Even though there's lots of space between the plants and plenty of opportunity for them to grow, they're just not thriving. And then over in the one corner of this field, there was a little cocktail crop, about double the size of this room. It had 12 species in it. And look at this cocktail crop. There's triticale in there, so the whole field was sown to triticale, and then they came back and they put another 11 species on top of it. So far more plants. Look how much green matter is growing there, and it's not being watered. It's not having anything done to it. It's just absolutely extraordinary that you can go from that to that. So that to me was, what is going on here? Like, how can this happen? Well, I've seen examples of this around the world now. Lots and lots of examples of this. When I was talking about this at the Grass-Fed Exchange in South Dakota last year, one of the other speakers, Brett Nix, came to my talk and then just spoke to me afterwards. He said, "What you're saying, Christine," he said, "we see it so many times." He said, "We had a really, really dry year in South Dakota last year. I planted a 27 species mix and a 5-way mix, and 5-way mix failed, 27 species mix never looked back." I said, "How come you put 27 species in a mix? Like, what made you do that?" He said, "Well, we've been planting these multi-species covers for about 5 years now, and we keep trying different combinations, but we only ever had 5. And he said, then we had in the shed, we had just odds and ends of seeds of all these different crops all left over. We decided to clean the shed up. We just piled it all in and just went, oh, well, we'll just put everything that's in the shed, we won't clean it all out and just throw it all in together." And he said, that's how come we tried 25 and we couldn't believe it, it thrived. Absolutely thrived in the drought, hardly any rain. And the 5 species mixes, he said, well, no wonder we've been having trouble with our covers. We didn't have enough diversity in there. And the Jena experiment in Germany, so they've trialled, this is a 15-year experiment. There's been several hundred papers written out of this. It's one of the most famous biodiversity experiments in the world. It ended, I think, in 2015. One of the things that they did in this trial was to look at comparing having 1, 2, 4, 8 or 16 different kinds of plants growing in these, well, 10-metre plots. I suppose that's about 10 yards, whatever. Sorry, I don't do imperial very well. And then they had none, 100 or 200 pounds of nitrogen per acre. So it's a multifactorial experiment. You could have one kind of plant growing in this area with none, 100 or 200 pounds of nitrogen, like all replicated, all fully replicated, right through to 16 different kinds of plants growing in one of these plots with none, 100 or 200 pounds of nitrogen. So you get combinations of all those different factors. I think it ended up with like 78 different plots or something. What they found was that if they had 8 or 16 different kinds of plants growing together with no nitrogen, it produced more biomass than having just 1 or 2 kinds of plants with 200 pounds per acre of nitrogen. That's nitrogen. That's not the product. I mean, if that was something like urea, which is 46% nitrogen, it'd be 400 pounds, right, of urea. So that's like, of actual nitrogen, 200 pounds per acre, could not produce as much biomass as 8 or 16 plants. They also looked at the microbes in those plots and found that, you know, where they had 8 or 16 plants with no nitrogen, they built far more soil carbon and had much more microbial diversity, much more life, and everything was healthier and the plants didn't get attacked by insects and pests, etc., etc. So there's a lot more to it. And soil carbon actually increased with increased species richness. Every extra plant type that they put in there, the more diverse they made it, the more carbon they built. And it was just a straight line like that, like going up at a 45-degree angle. Every extra plant that you added built more carbon. There's a great video on this if anybody wants it, I can send it to you. It's only about 8 minutes or something. They actually got 21% more carbon once they got over 8 species. They got 21% more carbon. So you can, like, the cost and everything would be pretty much the same. Just make it more diverse, you're going to build more carbon, and that's a really important point, I think. And the final example that I just wanted to give you before I talk about the science behind it was a recent visit I had in New Zealand last year. A dairy farm couple, Maya Smith, came to a workshop that I ran about 3 years ago, and I was talking about the need for them, the dairy farmers in New Zealand, to get off the high rates of nitrogen that they were using, because it's so detrimental to their soils, to their plants, to their animals, and to their waterways. The government's trying to legislate for people to stop using nitrogen, but they don't really have any answers. They're just saying to reduce it. I said to Maya, you could get off it completely by using a biostimulant instead of using nitrogen. And he went home and tried that on a small area and found that his milk production actually increased and that his animal health improved and everything. So he was happy about that. And he came to another workshop at the end of 2017 and was showing me photos of, you know, we've built this much topsoil, and he was really, really happy about his results. And I said, well, Maya, I've now got to tell you that you're not going to get any further than that if you just stay with ryegrass and clover. You actually have to have multi-species forages. And he goes, oh, I thought I had it all worked out, Christine. Now you're telling me that there's more. And I said, yes, well, we know more now that you can't really make much progress with a low diversity pasture. So he went home again and he put in a 2 hectare, 5 acre paddock. And this is the soil that he's working with. This is a pumice soil on the North Island of New Zealand. The North Island is all volcanic, but that doesn't necessarily mean like rich basalt soils. A lot of their soils are pumice. It's very abrasive, like when you rub it on your fingers like this, it's like, well, actually like using a piece of pumice. It feels really gritty. It's got virtually nothing in it. A soil test will show you there's virtually nothing in that soil. It's probably a bit like sand, almost like pure sand. The expert opinion is that you can't grow anything in pumice soils unless you use, like you have to apply not just NPK, but all the trace elements as well, you know, like zinc and cobalt, molybdenum, all of those things, boron, selenium. And so he was very reluctant to move away from fertilisers, but he found that he could by using a biostimulant. So now I want to show you what happened when he had a biostimulant plus diversity. So this is his soil. This is his ash soil. That's what it looks like if you don't do anything to it. This is what they achieved just by going to a biostimulant. His son dug that up and was showing it to me. So I'm now going to turn his son upside down. But I just want you to see the pasture in the background. So that's their ryegrass clover pasture. They were very happy with that because that looked heaps better than it used to look. It was just, when they're using all the nitrogen, it was just a horrible yellow colour. So it improved and they built that much soil. So if I turn Taylor's upside down now, but you just want to see the soil the right way up, this is how much soil, like say 6 inches of topsoil that they had actually built using a biostimulant. But you can see that pumice soil is still underneath. Now we went into this multi-species paddock. If you see those cars that are parked, so that you see the sheepdog here, like on the top of the hill, we're looking down, there's 5 acres there. Down in the corner you see those 2 white vehicles. They're at the corner. So everything else that's behind that is ryegrass. The trough, the water trough that you can see is in the multi-species paddock. So there's 12 different kinds of perennial plants in this paddock. And we just went in because one of the components was red clover, and I was curious to know, had it nodulated? Like having all those other plants around it, had it actually improved nodulation? So we just went in with a spade to look at that, like just totally not expecting to find anything really. And here we were just digging a hole to look to see. And what came out of that spade was this much black topsoil. And Maya couldn't believe it. His son is pointing to a little bit of pumice soil right at the bottom of the length of that spade. And he said, we must have burned a tree here. Like when he put the spade in, it just went woof, like straight in. And he said, and it came up and he goes, we must have burned a tree here. Like he didn't even react to the fact that it had come up with all this black spade, so, with all this black soil. So he went a couple of metres away and put the spade in again, and it went in really easily again and came up with all this black soil. And so then he went like over here and dug another hole, and it came up, and then he was like going around like a robot. Like he dug about 50 holes in about 2 minutes, and I'm going, if you keep going, there's not going to be any soil left in this paddock. And every single one, and he was just so totally stunned that he actually went to the pub and got drunk, like just totally blotto, because his dream had always been, I want to build topsoil. And the reason that he converted from not using nitrogen to a biostimulant was because I said, you will build some soil if you stop using nitrogen. And he was so impressed with, you know, his 6 inches of soil that he'd built. And when he saw this, like this is like almost unbelievable. In fact, I thought it was unbelievable too, because that multi-species cover had been in there for 5 months. He planted it in November the year before, and remember our summers are the other way around, so it grew through spring and summer, and this is autumn, fall, for us down under. So in 5 months of good growing season, and it rains in New Zealand almost every other day, so you've got everything like perfect for building soil. But the interesting thing is, with ryegrass and clover and nitrogen, you can't do it. Even though you've got plants and they're photosynthesizing and theoretically, you know, the liquid carbon pathway, all that, if you don't have microbial diversity under those plants, you're not going to rapidly build topsoil. So he went to the pub and got drunk, and then I went back to my hotel and I was thinking about it, and the next morning I rang him up and said, yeah, I need to come out and have another look, because I want to have a look around the perimeters of that paddock. For some reason that whole hillside could have just changed overnight or something, who knows. He said, I've been out this morning already, Christine, I dug another 100 holes, and I can tell you that, but I went out and I did have another look, and like just across the fence everything was like how it used to be, so I was happy about that. They've now yeah, so that's what he's done in 5 months. I mean, that's just, oh. So now we're at the top of that hill, looking out over the rest of his farm, which is all ryegrass and clover. And as I speak today, the rest of that farm now has all been converted to multi-species. And he said the same thing is happening in every paddock. But remember, he's already gone 3 years without nitrogen. Whether he could have done that, still using nitrogen, I'm not sure, because I'm going to show you some photos about what happens to plant roots when you do use nitrogen and what that's actually doing to the soil microbiome. So he's done all the right things, and he's also now increased his, what they call their round, what we might call a rotation. So he was on a 20-day round, so the cows come in every 20 days. I said, that's too quick. You've got to let that grow longer. Well, the theory is in dairy that if cows go into long grass, that their milk production goes down. And he's on a 45-day round now, sometimes extending that out to 50 days. He says every time he's extending the round, the milk production's actually going up. He can carry twice as many cows now. It's just like blowing his mind what he can do, and his neighbours are going, this is just against everything that we've been told. We've been told you can't grow grass if you don't use nitrogen. We've been told if you have long rounds and if you leave residuals like, you know, what they're leaving. He sends me photos of what they're leaving. When the cows have come out, they've taken half and left half and trampled it, and you know, there's manure all over it and everything. And I said, oh, that's microbial heaven. Your microbes will be so happy, and you look after them and they'll look after you. So we've been doing soil tests there as well. His cation exchange capacity has increased 50%. That's amazing in a pumice soil. That means it's going to hold nutrients instead of them going through in the heavy rainfall. All of the nutrients that we measured, including things like, you know, magnesium, calcium, sulphur, copper, zinc. The zinc just went through the roof. Unbelievable increase in zinc. Nitrogen and phosphorus increased. He's not using any phosphorus. He's not using any nitrogen. They've tripled. In his paddocks, his total organic carbon levels has tripled in the top 8 inches that we measured, and now that the soil it's down to a metre now. You sent me an email the other day, we're down to a metre, Christine. That's like a yard, down this far. So if you measured total organic carbon down to there, I mean, it would just be mind-blowing. Brix levels have tripled in his pastures. His milk production increases 300 litres every time the cows come through into that, what he calls his salad paddock. And the somatic cell count, which is looking at infection levels in the milk, has come way down. The cow fertility's increased 80%. I mean, what more could you want? And it's cheaper. It's actually costing him less, I think because the seed company didn't really know the value of those seeds, but it's costing him less to put in 12 species than to put in a tetraploid ryegrass, which is what they were using before. And he's not using any fertiliser anymore. So, like, you know, his costs are about 1/4 of what they were, and his milk production's gone up. His animals are more healthy. I mean, why would you not want to do this? So other farmers in New Zealand are doing the same thing, and we're finding across the board that increased plant diversity provided, you can't just put 3 or 4 things in. You've really got to get, I'd say like 12 is the minimum, really. improves animal nutrition, growth rates, milk production, conception rates, dependence on vets don't like that, and builds soil. So the title of today's talk was Community Tipping Points, Enhancing Nutrition Yield and Resilience Through Quorum Sensing. So what is quorum sensing? What is going on in these soils that explains these extraordinary results that we're seeing? It's definitely not a linear thing. It's like you reach a point and then bang, everything changes completely. So in human society, a quorum is the number of members of an organisation that must be present in order for decisions to be made or business to be transacted. So if we had like the board of No-Till on the Plains, or your local baseball club or something or other like that, there will be a certain number of people in that organisation that will need to be present for that group to make an important decision. So let's say it's the baseball club and you have to have 5 members or 7 members or something, the president, the secretary, the treasurer, and whatever board members have to be there in order to make a decision like, say, we're going to spend $10,000 to upgrade the stadium or something. If you have a meeting about that and only 3 people come, they can't actually make that decision. They have to say, oh look, we'll have to have another meeting. And if we haven't got 5 people here or 7 people here, we can't decide to spend that $10,000. And any of you who belong to groups, you know that you have to have a quorum to make those important decisions. Well, what we know about microbes is that they have to reach a certain critical density before they will actually initiate any kind of action. And it's really interesting to know why they do that and how they do that. So in the microbial world, which is the world, remember, we're a tiny, tiny part of biomass on this earth. We live in a microbial world. The term quorum sensing, which is usually just abbreviated to QS, refers to density-dependent coordinated behaviour. In other words, you have to have a certain number of microbes and then they all work together like a superorganism. It's coordinated behaviour and they can regulate gene expression. Bacteria in your gut can switch your genes on and off depending on how many of them there are and what kind they are. You don't get any control over that. It's really amazing. The bacteria in a plant can switch that plant's genes on and off. The bacteria in the soil around a plant's roots can switch that plant's genes on and off. And what we know is that we don't have enough bacteria of different kinds in our guts. We don't have enough, it's not just bacteria, it's also fungi and archaea and viruses. The viruses are incredibly important. So the whole microbial life, I suppose I should say, if we don't have enough of it, there are certain genes within our plants, let's just concentrate on plants for the moment, that they need for tolerance to drought, for example. Look at that amazing drought tolerance we saw, for tolerance to frost, to tolerance to pests, to tolerance to diseases, and for growth. And if all of those genes are getting switched off because there's not enough microbes there to activate them, then you've got this poor plant. It actually has a genome that's capable of doing all kinds of things, and most of the genes have been deactivated, just like they have in our bodies. That's why we're getting so many autoimmune disorders, because we just got our immunity genes have just been switched right off. So it's density dependent, coordinated behaviour. It occurs in all of those microbial groups. And the more we learn about it, the more we realise just how potent it actually is. So why does it work like that and how does it work? Well, if you think about it, microbes, there's a lot of them. They can't see, they can't speak to each other, they can't hear anything, so they can't communicate like we do, but they do communicate extremely well and they're incredibly well organised. I'm going to show you some examples of organisation of microbes and what they can do. So how do they do that? Well, every species of microbe produces its own unique signal, which is called an auto-inducer. And when the concentration of auto-inducers in the environment reaches a critical level, then that's when they can either change their genes or they can change the genes of their host. And one example of this is a marine bacterium called Vibrio fischeri. When there's just a few of them in the ocean, they don't do anything special. When you reach a certain critical density of them, there's enough auto-inducers in the water for them to determine, they can't see how many of them there are, but they can tell that there's a whole heap of other ones of the same all there. at night in the dark, they switch on their genes for luminescence and they become bioluminescent. So they shine in the dark. It's all this beautiful blue. Don't know whether any of you have ever seen those blue phosphorescing bacteria that you'll see sometimes if you're in a boat at night. You'll see all this beautiful blue colours in the water coming off, and these are bioluminescent bacteria, but they only produce that blue luminescence when there's a certain concentration of them in the water. And that's really interesting to know why they do that. But what we're seeing in the soil is that you have to reach a critical density of bacteria or fungi or archaea or whatever it happens to be for certain things to happen, just like a quorum in the human population. So they have their own language, but they're also multilingual. They are very clever. They may be small, but they ain't stupid. So they have a species-specific system that says me, how many of me there are, if I'm some kind of a bacteria, and then running parallel to that, they have a second language that is interspecies. So if you've got lots of different plants growing in the soil, the bacteria and the fungi and the archaea and the viruses that are in the soil know through these chemical signals that there's a whole lot of plants there. And if there's a whole lot of plants there, they respond differently, just like a quorum in human society. It's quite amazing. They know how many of us are there and how many of them, like how many other things are out there. And they use that information to actually decide what they're going to do. And if you think about our bodies, like right now, you're sitting there, hopefully listening to me, but you know, your heart, liver, kidney, spleen, adrenals, you know, thyroid, hypothalamus, everything, all those different organs in your body, they're all communicating with each other using biochemical signals that you're not aware of. And if they weren't coordinated, you wouldn't function as a human being. So if we interrupt those signals somehow, or don't have enough of those signals, then things are not going to work properly. And I mean, we, at a conscious level, know when we're hungry, we know when we're tired, and a few basic things like that. There's a whole lot of other stuff going on. We don't really know at a conscious level how well our kidneys or our liver or whatever is functioning. But your body knows, and you know, blood tests and things like that will tell you. So it's really no different. So we're one thing, a unit, but we've got lots of different organs that are all connected through this signalling mechanism. And we've got heaps and heaps of microbes in us that actually outnumber us, as David was talking about and Anne was talking about. You know, on a cell count, we're way outnumbered. On a gene count, we're just like totally outnumbered with the microbes that live in and on our bodies. And so when you think about the plant, it is also going to be outnumbered by the microbes that are living, well, hopefully outnumbered by the microbes that are living around its roots. And those plants, those microbes are going to be able to send signals that can actually switch genes on and off in that plant and make it either more or less productive. So a plant or animal genome is the complete set of genetic material in that organism, including all the viruses and bacteria and everything that live in there. And a lot of that DNA has up till now been considered to be inactive, but we know that they actually can be influenced by quorum sensing. So if we were just to look at this in very, very general terms, like just forget about all that detailed biochemistry for a minute, we know, like just by looking, that above ground plants provide habitat, they provide food for other life. I mean, we can see that when you've got something growing above ground, it creates a whole dynamic of life, like a whole lot of things that can exist. What we tend to forget is that, and we know that, you know, if we eliminate plants, then there is no other life, really. Not very much, there's a little bit, but above ground, not much is going to happen. We now have to think, okay, you know, underground, if we've eliminated plants, there's that whole other half of life that's underground. We're going to disrupt everything. And I think, you know, people, especially in this room, you understand that very well. And you understand very well that the presence of year-long green diverse ground cover is the most important factor for soil health. I think you've heard that so many times over so many years that you've actually got that. But it's because those living plants actually support the microbes that create that well-structured, fertile, friable topsoil that we want to have. The plants themselves can't make the soil. The plants can produce exudates that support the microbes, but it's the microbes that actually form that soil. So it's not really surprising that if we get more microbes and a greater diversity of microbes, and if quorum sensing comes into the picture, people go, oh, how can you suddenly build that much topsoil? But when you realise that what you've done is activate those microbes and they're the ones that are doing it, the plants might be just exuding the same amount of carbon, but what are the microbes doing with it? So their carbon use efficiency is just increased incredibly. It's not being wasted like it was before, or not so much of it's being wasted. David talked a little bit about carbon use efficiency in his trial, showing that if you had more fungi in the soil, you could increase carbon use efficiency from, I think, 11% in a bacterial dominated soil to 55%. What he means by that is if you had the same amount of carbon coming in as plant root exudates in a bacterially dominated soil, 11% of that might remain in the soil as stable carbon, whereas 55% of that will remain in the soil as stable carbon in a fungally dominated soil. Like, that's a five-fold increase in how much carbon actually remains from the same amount coming in. That's huge. And that's the sort of thing that is going to build soil like really quickly. How can you tell if soil building's happening? Well, you'll see these riser sheaths around plant roots. They're the microbes that built those. So that's incredible engineering feat when you think that somehow or another, these little things that can't think, see, hear, talk, can't communicate any other way other than chemically, have got themselves organised to build riser sheaths around those plant roots. And inside those riser sheaths, there's all kinds of extraordinary biochemical transformations taking place. They can basically access anything that they need for that plant, including atmospheric nitrogen. So any kind of plant, doesn't have to be a legume. Anything that's green and photosynthesising and has a microbial community around the roots that can form riser sheaths can fix nitrogen. There's a lot of nitrogen fixing happening. It's the same as what happens in a nodule on a legume, but this is much more general, and any plant can do this. If the roots are clean, like those ones in the bottom, they're only clean because plant was just yanked out of the soil like that. Otherwise, they would have had riser sheaths as well. Clean roots on plants means it's not talking to the soil microbiome, it's definitely not forming a quorum, and it's definitely not doing any of the things that we would like a plant to do. This is a photo that David also showed in his talk. This is from Ian and Diane Haggerty. Jay Fura mentioned the Haggertys using a vermi liquid on their, well, he said they were putting it in the furrow. That's not technically correct. The vermi liquid actually goes onto the seed before planting, and so you can see this little seed on your right-hand side it hasn't even got leaves yet. It's just produced 3 seminal roots and a coleoptile. It hasn't broken through the soil surface yet, and already look at the microbial, what the microbes around those plant roots have. Look how much energy it has taken for them to produce the glues and gums to pull that soil together and form riser sheaths around those roots. I mean, that's extraordinary. This plant's not photosynthesising yet. It's not moving liquid carbon down into the roots. It's like this is all just signalling between microbes and that seed as a result of that inoculant going on that seed, and that's where it needs to go. If you take a seed and surface sterilise it and place it in the soil, it doesn't even know which way up is. It will grow leaves downwards and roots upwards, or leaves and roots sideways, because the only way that it knows which way is up and which way is down is through what we call a geotrophic response. It can detect gravity and can respond to gravity by growing leaves away from it and roots towards it. And that's a hormonal thing. So if it doesn't have any bacteria on the seed coat, all its hormones get switched off and it gets totally confused. So the more auto-inducers, more chemical signals, beneficial ones, that we can put on the outside of a seed, it thinks it's growing in one of the most fertile environments in the world because it's getting all these chemical signatures coming through the seed coat, and goes, oh wow, I'm growing in this, and I'm just going to grow amazingly because I'm growing in an amazing place. So it could be out in the middle of the desert and it thinks that it's, in a rainforest or something, because of all the chemical signals that are coming to it. And look at all the soil sticking to that plant that's on the left-hand side. Now if we go and look inside this riser sheath, this is what we see. So on your left-hand side is the edge of the plant root, and then on the right-hand side you've got the soil particles that are all being stuck together, and you can see how they're being glued to that plant. Look at all those fungal hyphae. You can see little drops of liquid there, the root exudates, like the liquid carbon pathway, and there are going to be just like thousands of different kinds of bacteria and archaea and viruses, like beneficial viruses, which every bacterial cell and every fungal cell and every human cell, if we don't have beneficial viruses in there, they don't function. We just realised that the human virome, the plant virome, the soil virome, it's actually more important than the genome. That's going to be one of the big things in our future research, we're going to start looking at viruses and at beneficial ones and how they work. Just like a few decades ago, we thought bacteria were bad and we thought fungi were bad and we thought nematodes were bad, and now we realise that the beneficials outweigh the, the detrimental ones by a huge number. Same with viruses. So in that incredibly diverse and rich, very protected environment inside that rhiza sheath, the biochemical transformations that are taking place in there, the signalling that's going on, there are genetic changes taking place in that plant as a result of the signals it's getting from that rhiza sheath. It doesn't have that environment around it. It just thinks, oh, I'm growing in a really sterile place, I'm just not going to grow. And if we come back a little bit, this time this is only 300 times magnification, we can see how those fungal hyphae are pulling soil particles together and making those aggregates that's going to give you a soil structure and allow water to infiltrate and all the other good things that we want the soil to do, as well as grow plants. If we just look at this diagrammatically, the one on your left is unstructured soil, it's just going to be like a brick, and then the one on the right is where it's been aggregated by microbial activity. That's going to allow air to move through, water to infiltrate, plants' roots to grow easily, things like earthworms to move around. That's the holy grail, to have that well-structured soil, as you all know. And we now know that plant root inputs build soil carbon 5 times faster than any kind of carbon derived from above ground biomass. But the thing is, they cannot do that if we're using high analysis fertilisers. So you could have all the diversity in the world and all the green that you want, and if you're still using nitrogen under the seed, I'm sorry, this is not going to happen. So this photograph here, I haven't got a pointer so I can't show you, but you can see the roots, and if you go about halfway down through the root mass, you'll notice that there's 2 wheat seeds there, and then the roots that are coming out from those seeds are totally bare, naked roots. Why is that? That's because this farmer put 80 units of N in under that seed. The university people recommended that they do that. That's pretty amazing that the plant actually even grew at all. But it has just basically sterilised the soil around those plant roots. Now, if you look at the roots that have come from the crown of the plant, they've all got rhiza sheaths, so they've got out of that zone of the nitrogen. Normally what we see is where people have used nitrogen under a crop, all the roots are just going to be bare, like those bottom ones. But the reason that there's roots growing out of the crown was that they came along with compost, actually had compost put on the surface, and that changed the environment of that surface soil. So the roots that grow in the surface had rhiza sheaths, and the roots that grow below the seeds didn't. And for me, that was a classic example. If you ever wanted to be convinced that nitrogen's not a good thing to put under the seed, this experiment convinced me. So again, this is the Haggerty soil, the people that are using the vermi-liquid that Jay was talking about. This is what it looks like. You can't even see the roots of their plants because they've got all this soil aggregated around. We jumped over the fence and took a sample from the neighbour who uses nitrogen under their wheat. So everything was like the same age, same stage of growth, soils are the same day. And this is the neighbour's roots, what they look like under high magnification. There is no communication with the soil. So the neighbours' crops all had, fungal diseases on the leaves and all kinds of things. They're having to use a fungicide. They didn't grow very well, so they kept putting more and more nitrogen on. And how many places around the world do we see that kind of thing replicated? We're just not understanding that what we need is a really fertile microbial environment around plant roots and diversity and biostimulants. The reason I think biostimulants work, like when you put them on the seed, is there are so many chemical signals in there that come from the whatever method has been used to produce it. It's not the microbes themselves, it's the auto-inducers. But now that we know that the way microbes communicate is to produce auto-inducers, if you just take all the auto-inducers and put them on the seed, it's like it thinks it's growing in a compost heap or whatever it might be, because that's all the signals that are coming to it, and goes, oh wow, I'm in a great place, I've got lots of microbial friends, I'm just going to grow like mad because they're all going to support me. If we just look historically at what's happened over landscapes, North America, Australia, South Africa, some parts of South America as well, we've gone from prior to European settlement having nice healthy soil with green ground cover, so that's on your left hand side, and then the next little section is net primary production decreased. In other words, we've lost ground cover. Could have been through overgrazing, could have been burned, could have been sprayed out or ploughed out. Whatever way we've chosen to annihilate the ground cover, the next thing that will happen in the next section is soil organic carbon decrease. We're going to lose carbon if we lose ground cover. Bare ground always loses carbon. Bare ground always loses carbon. And then when we lose carbon, the final section, soil moisture decrease. If you don't have carbon in the soil, it's not going to be able to hold moisture. Then we have dramatic increases in evapotranspiration. And in fact, there's now a lot of theories going around that this is the main source of global warming, that we are getting warming from loss of moisture from the soil surface because we've bared the soil surface, that we need to go back now to ground cover to actually stabilise our climate. So we have caused an increase in the frequency and severity of droughts through inappropriate land management. When you consider that the huge areas, the millions and millions of hectares around the world that have been undergone this kind of a transformation, it's orders of magnitude greater than the tiny effect that we get from doing something like burning fossil fuels. So it's inappropriate land management that is destabilising the climate, and we have to think about how plants, soils, microbiology, the hydrology and global climate, none of those things can be considered in isolation. Just like we can't be considered in isolation from our microbes, our soils can't be, global climate can't be either. Because all these things are connected, and when we look at what's changed since European settlement in the major continents on the planet, we've incredibly simplified the landscape, we've incredibly reduced the amount of green, we've incredibly reduced the diversity of plants. The organic carbon content of nearly all of our soils, agricultural soils, are 50 to 80% less than the original level prior to settlement. And that is so important for water holding capacity, for the nutritional status of plants. And therefore it's going to be a very important factor for farm profit, because all of these people that have discovered ways of rapidly building soil carbon are far more profitable as well, because they don't need the inputs anymore. As a result of all these things, soil fertility has climbed. It's now dry when it was once hydrated in many places. And then what happens when it rains? The water sits on top of the ground because it can't get in. We don't have good soil structure, good infiltration, all those pore spaces, all those aggregates creating spaces for water to get down. It's going to sit on the top of the ground. And when it sits on the top of the ground, rather than infiltrating, we get an increase in water vapour in the atmosphere. And water vapour is the greenhouse gas that's increased to the extent since the Industrial Revolution, by an order of magnitude compared to carbon dioxide. And it's a scientific fact. If anybody wants the information, we've certainly got it, that water vapour accounts for 95% of the greenhouse effect. And we've known this for a long time, that water vapour has been far more significant than carbon dioxide, and I don't know why it's been ignored. It's not new information. It's just that I think people thought, well, we can't do anything about water vapour, but we can. With appropriate land management, we can do a huge amount about water vapour. So carbon dioxide in the air, 3% of the carbon dioxide that's being emitted to the air, or that's increasing in the air every year, comes from the burning of fossil fuel at the most, and carbon dioxide constitutes 0.04% of the atmosphere. So if we're really serious about reversing climate change, we need to put the water back in the soil where it belongs. That needs to be the mantra, put the water back in the soil where it belongs. But if you're going to put the water back, you need to have the carbon to hold it. So we've still got to put carbon in the soil to hold the water, but it's the water vapour that we want to get out of the atmosphere. And the other thing is that people go, well, if there's so much water vapour up there, how come it's not raining more? And the answer to that is that, sorry, just looking at the clock, I realise I've got 2 minutes left. The answer to that is that the water vapour, it just forms like a haze, and it doesn't actually nucleate unless we have bacterial, usually bacteria, what are called aerosols. It's a funny word for them, but bacterial aerosols that actually form a nucleus for either rain or for snow. So it can't coalesce into drops and fall as rain unless it's nucleated with something. And where do those bacterial aerosols come from? They come from green plants or coral reefs. If you notice, like if you look at where it rains over the ocean, it rains over coral reefs, heaps more than it rains over other areas where there's no coral. So there's lots of bacterial aerosols come out of coral reefs, there's lots of bacterial aerosols come out of green plants. So if we have ground cover, Well, we're going to reduce evapotranspiration hugely for a start, and we're going to produce bacterial aerosols that can nucleate that water vapour haze that's up there and coalesce into raindrops, and it might start raining again in places where we need more rain. So we need to understand how to harness this power of microbes, how to use quorum sensing to our benefit. It's going to produce food of high nutritional quality, because all those elements that are becoming available in the soil are obviously going to be in our plants, which is one of the keys to human health. But it's also going to dramatically improve water retention in our soils, which I believe is the key to reversing climate change. But we need a quorum of people to actually realise, understand all these interconnections, because until we get a quorum of people that understands these things, then there's not going to be any change in our society in response to these. Once we actually get that understanding, then we can restore human health and climatic stability. So again, to come back to the title of this talk was Community Tipping Points, Enhancing Plant Nutrition, Yield and Resilience through Quorum Sensing, but that applies to people too. So as you know, with all the sorts of changes, the innovations that we've seen, going from full cut cultivation through to like zero till, and then cover crops, and then diverse cover crops, and then weaning off fertilisers, and now looking at biostimulants and all of these things, we have to get to sort of tipping points in the human population before these become accepted as ways of going forward. When there's only one or two people doing it, they're regarded as being a bit strange and on the outer. And then when we get to a point where everybody kind of looks back and go, wow, did we really do that? Did we really plough up all of our soil? Did we really use all those dreadful high analysis fertilisers and all that insecticide and all that fungicide, and did we really put all that poison on our food and then give it to our children? I mean, did we really do that? So, we get to a point where we look back and can't believe what we used to do in the past. Well, we need to, understand a little bit more about how the global climate works and about how microbes are really running this place, and we need to figure out how to work in with those to get it to work better for human health and for climatic stability. Thank you.